Welcome back. So today we move into genetically modified organisms, a brief digression into genetics and society. Hopefully, if you just walked in, please take this one question Socrative quiz. Well, two questions really, but we're going to see, once you add, should foods have to be labeled as being genetically modified? Okay. We're going to see what the class opinion is. So we've got 58% of you saying yes, 24% say it depends, and about 17% of you say no, foods shouldn't have to be modified or labeled as genetically modified. So overwhelmingly, sounds like yes. Although some of you answered yes, sometimes it depends. So I didn't know multiple answers were possible, but they are. Here's an advertisement I took out of a newspaper I got a year or two ago from one of our local suppliers. It's a local grocery store, not Fresno State, fortunately. What's interesting about what they've written here about their tomatoes? I'm not going to tell you which grocery store this is either. Where do they talk about being genetically modified? What definition have they basically told us is their definition of being genetically modified? So what have they excluded as genetic modification? The way they've written this. Crossbreeding and hybridizing. So they say, our suppliers have been crossbreeding and hybridizing tomatoes for years, but they're not genetically modified. To me, as a geneticist, this seems like a counterintuitive statement. And so one of the first things that I'd like you to do, after we do this, is we're going to get into groups and come up with your definitions of what it means to be genetically modified, and then we're going to discuss that. First, though, I wanted to make sure that everybody saw my version of the answer from last week's Socrative exercise, drawing the genetic pathway related to the GAL pathway in eukaryotes. These are two student-submitted responses from two of you, and these are accurate, but not necessarily the only accurate answers to this question. They both represent essentially the same pathways. Two of you came up with the same response. So let me draw this out for you quickly in case somebody has questions. So what I'd like you to do while I'm drawing out the molecules involved, look at these two pathways and see if you have complaints, concerns, or questions about the two pathways that have been proposed here. What's the gene that this pathway regulates? Is it GAL4, GAL80, GAL3, or GAL1, that gene that gets turned on in response to the presence of galactose? This happened to be GAL1. It's okay. So what protein binds the upstream activating sequence? There's a specific DNA sequence that's an enhancer for turning on GAL1. Which protein binds there? This is GAL4. Now, what protein is sitting on GAL4 that keeps it from turning on transcription? This is GAL80, yes, in the back, absolutely. So GAL80 binds to GAL4 and prevents it from turning on transcription. So when the galactose sugar is present, how does the cell get GAL80 off of GAL4? That must be the other players in the pathway. That's GAL3. And in what circumstance can GAL3 remove GAL80 from GAL4? Presence or absence of galactose? Right, presence. So together, GAL3 and galactose, what do they do to GAL80? Do they blunt arrow or arrowhead? Does gal-3 activate gal-80 or repress gal-80? This is where it gets mired down in detail, and that's why if you have a question, please ask. So the definition of activation or repression is sometimes very vague. And it might even seem like it depends on your perspective. What's gal-80 doing to gal-4 right now? Is it activating or repressing? Right, it's blocking gal-4 from interacting with RNA polymerase. So gal-80, it is definitely blocking or repressing gal-4, and that was something that was common to both of these pathways that these students suggested. If gal-80's normal job is repressing gal-4, and gal-3 removes gal-80 from gal-4, then is gal-3 repressing gal-80 or is it activating gal-80? gal-3 is preventing gal-80 from doing its normal job. So it's repressing. Together with galactose. Galactose plus gal-3 are repressing gal-80. So what happens if you repress a repressor? The double negative. If you've got galactose present, gal-3 represses gal-80, which represses gal-4. That basically means double negative means positive. There's an activation. In the presence of galactose and gal-3, gal-4 is activated. And then it can bind to RNA polymerase and turn on transcription of gal-1. So both of these pathways reflect the way I would have drawn these genetic pathways. Does anybody have a modification they would make to those two pathway drawings? Anything that's not clear or that you would actually argue vehemently against? Somebody better.
What produces the galactose? Was it the gal-1?
So galactose, the sugar, is supplied, exists in the medium. So it's only when these cells, for example, are growing in the presence of that sugar that galactose is present, and that turns on transcription of gal-1 through this pathway. Does that satisfy the answer, or do you have a different question? So galactose, the sugar, gets into the cell. The cell can use this pathway to recognize whether or not galactose is present. So it binding to gal-3 removes gal-80 from gal-4, and then transcription proceeds.
Would it be accurate to draw a second arrow from gal-3 going to gal-4?
So could you draw, on this one on the left, for example, that, if I understood the point correctly? It's true. Gal-3 does indirectly act to activate gal-4, but it does it through repressing a repressor. What about this? This pathway and this pathway both show galactose activating gal-3. And then gal-3 represses gal-80. What would happen if on the version on the right here, what happens if you get rid of galactose in this pathway diagram? So say the cell is not in the presence of galactose. Does it want to transcribe gal-1? No galactose, right? You don't want transcription of gal-1. But what's this pathway going to do in the absence of galactose? What's gal-3 going to do? The way this pathway is drawn, with gal-3, it would repress gal-80, which would not be able to repress gal-4. So what I would prefer doing, instead of putting galactose upstream of gal-3 as an independent player in the pathway, I would probably put gal-3 plus galactose together repress gal-80. So gal-3 and galactose would be at the same step in the process, which makes it clearer that if you got rid of either one, either if you were in the absence of galactose or if you didn't have gal-3, there would be nothing to repress gal-8. So those two molecules, the sugar and the protein, both work together at the same step in the process of repression. Other questions or concerns? So, concerns about genetically modified organisms then. Is this going to happen to you if you eat a genetically modified organism? I don't know. Depends on your definition of what a genetically modified organism is, I guess. So the first job then is going to be to get into small groups, briefly, maybe 5 minutes or so, come up with a consensus, 2 or 3, maybe 4 of you in a group. What does it mean to be genetically modified when we're talking about genetically modified organisms? To you, try to come up with a good definition for it, and then we'll run through a few examples of genetically modified organisms. So what I'd like you to do is to get in these small groups, and then post just one of you, one person from each group, paste the text answer, your definition, onto that website, and then we'll collect everybody's definition there. So what does it mean to be genetically modified? Come up with as precise a definition as you can. Posting if you're not quite done yet. So genetically altering a DNA sequence to produce the outcome that you want. Right? An alteration that would not commonly occur in nature. So, not commonly occur. Okay. Why is it the commonness of the mutation that would be important, or the genetic change?
Maybe you're picking the genes and you're concentrating just on that type of, that expression. So the more common, I don't know, there's more of that kind.
Okay, so it's, yeah, if it's common, does that mean it's genetically engineered, or does that just mean that it's common? A change in the genome of an organism, so let's see, a change in the genome of the organism that changes its traits. Isn't that just mutation? A mutation could change the trait of an organism that's changing its genome. Combining two organisms that would not breed in nature. Can everybody agree to that as a definition of genetically modified organism? You get a combination of genes from two different species that can't make offspring in nature? Selective breeding doesn't count as GMO. Wait a second. So top one right there. An organism that has had its genetic material altered in a way that doesn't occur in nature. Feelings? Thoughts? Does GMO have to occur in a laboratory? Is that sort of an inherent... When I think of GMO, I think of, scientists with micropipetters and... Who says no?
And what about like domesticated foods?
Okay, what about domesticated foods? So everything pretty much is genetically modified. I mean, as Dr. Constable was leaving just before us, he said every one of us is genetically modified. We're a unique combination of mutations passed on by our parents. So how do you define what a genetically modified organism is? Does it have to be something that was derived in a laboratory? Is it something that combines genes that could never be combined together in a single organism? So let's play Spot the GMO. What's in the upper left there? A giant tomato, and just underneath the finger is a little tiny tomato. Genetically modified or not?
Yes.
Okay, what's the basis for the difference in size between these two tomatoes? Are they genetically identical or not? No, they're not genetically identical. There's a genetic difference that causes that difference in size. It's not like that's a baby tomato and that's an adult tomato. These are two ripe tomatoes. They're genetically different. That genetic difference arose in nature. They're two different subspecies of tomato. So does that count as genetically modified or not? Naturally occurring mutations that cause different tomato cultivars to have different sized fruits. Does that count as GMO? What about down here? What is this picture at the bottom right?
Grapes.
Grapevine. What's that little ribbon doing there on the trunk? It's a graft. Genetically modified. One organism that now has two different genotypes. You've got the rootstock, and you've got whatever grape variety was grafted onto the rootstock. Is that genetically modified? It's one organism now, part of one plant stuck onto a part of another plant. Does that count as genetically modified? What does this t-shirt represent? Yeah, corny. Right. So the first question I want you to think about is, how would you know, just by looking at it, whether or not any of these foods should count as, depending on your definition, there's no one definition of genetically modified, so you all come up with some definitions. Is this a genetically modified watermelon?
No.
Why not? Yeah, you guys have, you know too much. Okay. Right, so this is, but the point is, if you just look at an organism, it's hard to tell whether or not nature caused the difference you see, or if it was genetics. Right? Is it genetics or is it the environment? Is it nature or nurture? In this case, right, you can buy, what is that, melonmold.com? You can buy these clear acrylic boxes, you put them around the fruit as it's immature. It grows up, it fills up the box, and then you get square pumpkins, square watermelons, right? Not genetically modified. That's nurture. But what about the grapery from Delano? Have you guys had some of these grapes? The cotton candy grapes. They have these grapes that have interesting flavors and interesting shapes. Do you get the witch finger grapes like shown here in the gentleman's hand? Really long, skinny grapes. But then look at the flavors over here on the left in the menu. We got the flavor promise grape, the cotton candy grape. I've had one of these. It actually does kind of taste like burnt sugar a little bit, the caramelized sugar, cotton candy grape. So a local company has been developing these grapes that have different flavors. Is this GMO or not?
Yes.
Well, they claim, wait a second, they claim here, right here, non-GMO. Right there on their labels. So what did they do to make these grapes? Taste like cotton candy. Yeah. Pardon? They bred them that way. Selective breeding, the way that a lot of farmers have been doing for generations and generations and centuries. You select offspring that have the characteristics that you want, you breed them with similar organisms, and you breed in these traits. So they don't do anything in a laboratory to make grapes that taste like cotton candy. It's just over many generations they've found grapes that kind of taste like cotton candy. You breed them together, they taste a little bit more like cotton candy, you develop these certain flavor profiles. So we're not going to arrive by the end of class at a definition of what a genetically modified organism is. I just want you to be thinking about your own definition of genetically modified and whether or not these fit your definition or not. Should these be labeled as genetically modified? Conventional breeding, does that count? What do you think? Should we disclose when crops are conventionally bred? Yes or no? Does anybody say yes? Okay. Do you mind telling us why you think that? I'm not disagreeing.
You have a right to know what's in it.
Okay.
Whether it's in it or not, they should just say.
Fair enough. So absolutely. So we shouldn't draw any lines in the middle. We should say you have the right to know what's in your food, and so all the information we might want to have should be available to us. We could label things as conventionally bred versus GMO, maybe. I'm going to talk a little bit about society and the legal system and how it gets involved as well, briefly. I know this is not a law class. So here's a statement from the grapery on how they get their grapes to be these wonderful flavors. I know you can't read it up here. Hopefully on your tablets you can. Look at what they say in the answer to, are your grapes genetically modified? Right. as was suggested, the gentleman in the back, no, they work hard to breed their grapes naturally over time. Okay. What's the answer to the next question up here? May I please have some of your plants to grow my own? What do they say? Hell no. They're ours. They're patented. Does that sound like genetically modified or not? There's some proprietary interest in how those grapes have been developed. There's a particular genotype, right? There's a particular DNA sequence in these plants that they're trying to protect. To me, that kind of sounds like something proprietary. It's conventional breeding, yes. But when you start involving the law, it makes me start thinking, I don't know. Maybe there's something to this. Does it count as genetically modified or not? I already mentioned grape grafting, right? If there's any sort of frankenfood, it's going to be, like Frankenstein, something that's had multiple parts grafted onto it. Right? When people complain about, I don't want to eat frankenfoods. Apples, grapes, lots of plants get grafted. You get lots of varieties, citrus. You get a pest-resistant rootstock, but you get grafts on it that grow delicious fruit. Does grafting count as genetically modified organism? Sir?
No, there are two organisms that have not been altered in any form way, they're just junk. Actually modified, they're just symbiotic, I guess. You know?
Oh, so there's a relationship between the two, but you don't count them as one organism. There's two organisms that are growing together, one growing on the other. But it's an interesting point. So neither of these, until recently distinct plants, has had its genes changed, i.e. not genetically modified. So what should count as genetic modification? Does anybody have an example of a genetically modified organism that comes to mind? Yeah? So banana is genetically modified. But then bananas have had that problem for a long time before we were able to genetically modify plants.
I know there was a tomato where they tried to make the skin firmer, so they used a peanut gene. I feel like that would be genetically modified.
Okay, so now we get into this idea of taking a gene out of one species and put it into another species, which seemed like that was sort of a basic distinction between a lot of the species or the genetically modified organisms concepts that you put up here on today's meet. That if you're moving DNA from one species into another without the traditional process, that being mating, breeding, maybe that is a good boundary to put on what's genetically modified and what's not. Emily?
Seedless grapes.
Seedless grapes. Was that genetic modification?
It's crossbreeding two
So, crossbreeding again. So what about inserting DNA from new sequences, or from new organisms? Any examples of that, Jason?
I know there's one that's been popular recently called the Arctic apples. It's the apples that don't brown. I know they get it through your plasmids.
Right. So we're going to get to, yeah, absolutely. The Arctic apple, we're going to get to in just a minute. It's a great case study for what you might consider the most genetically engineered, but maybe not. So back to the point, just briefly, about whether or not we should be required to label genetically modified organisms or not. I just wanted to give you a brief taste of what's happening in policy right now. A year and a half ago or so, the Safe and Accurate Food Labeling Act passed the U.S. House of Representatives and is currently in the committee in the Senate. So it's not a law yet, right? One branch of Congress has passed it, the other branch is still talking about it. If it was passed by the Senate, it would just prevent states from passing their own laws. So it would reserve this decision to be made at the federal level, whether or not you'd have to report whether or not your food was genetically modified or not. And it doesn't get into the details that we're discussing now of what counts as a genetically modified organism. So right now there's definite interest in the federal government at trying to figure out whether or not foods should be. why wouldn't corn count as a genetically modified organism?
Its natural ability to adapt or evolve within the time frame that is given.
So something about its ability to adapt or evolve in a time frame. So how long did it take the ancestor of corn, teosinte, shown here, to turn into what we know as corn or maize? It's been hundreds of years, if not 1,000 or more. So a long period of time it took to go from teosinte, the wild ancestor of corn, to corn. And if you mate teosinte and corn together, this is what you get. Right? You get teosinte, you get corn, and you get an F1 hybrid. It looks very much like you'd expect an F1. It sort of looks like halfway between corn and teosinte. There's one genetic difference that makes teosinte useful as a food source. So the problem with teosinte, which is, again, the ancestor of all corn, is it's got really tough kernels around the fruits. So it's like tooth-crackingly hard. It'd be really hard to use teosinte as a food source. And we know that domestication occurred in central Mexico, and by dating the starch fibers that are found in these tools that were used to mill the corn, we can date the origin of modern corn to about 8,700 years ago. So it's been relatively recently, but still thousands of years ago it was said, right, that maize was domesticated. So it's been a while since maize evolved, thanks to us, from teosinte. So this is, again, teosinte on the left, B, with the closed fruit cups. They're really hard to access the nutritional value to us from the organism that's shown here as B. And there's a single mutation, one genetic mutation that makes teosinte look a little bit more like C. The fruits are a little bit more open. And that's the trait that humans selected on those thousands of years ago to develop teosinte into what we now call corn. And that gene that this mutation was in is called TGA1, teosinte gloom architecture 1. So this is a fabulous story that was really widely heralded back in the mid-2000s when this was discovered. So a single mutation could be responsible for a major transition in a food source, like a critical food source for humans. So this is how this study proceeded. If you want to find the mutation that causes a difference between teosinte and corn, what do? What sort of information do you need? If you're looking for a change in a DNA sequence, what do you need to collect? DNA sequences, sure. So then we have a large number of DNA sequences. Across the top here, this first line, maize, this is the DNA sequence. Not a continuous DNA sequence. So here we're looking at position 1016, 1014, 1012, and so forth. So we're looking at a few nucleotides across one section of one maize chromosome. And every time there's a dot, that means that that second maize line had the exact same sequence. So when you see dot, 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 that means that maize line MH2 had the exact same sequence as the one that's shown above it. So let me draw a line here. You do the same thing. Draw a line between where it says maize and where it says teosinte. What's true about most of the maize plants in their DNA sequence around this gene? Are they different or are they this? They're almost identical. So you take lots of different maize lines, accessions, varieties. You sequence their DNA. They're almost all identical, except for where? What's the least identical part of the different maize lines? What was it? 951. There are a few of these lines where instead of having a G, most of these lines have a G there. But there are 1, 2, 3 that have a T instead. Below the line are a bunch of different teosinte, the wild ancestors of corn. Their DNA sequences of the same gene. And what these authors were looking for was, what is the genetic basis for the difference between open kernels of corn and those tightly encased ones like teosinte has? Is there one spot in this figure where all of the teosinte individuals have one nucleotide and all of the maize individuals have something else? So we're looking for a correlation between the phenotype, open kernel, closed kernel, and genotype. Does that exist? Where does every teosinte have the same nucleotide and none of the maize plants have it? It's the last column, right here, position 18. Every single maize plant has a C there, and every single teosinte has a G. So if you were looking for a perfect genetic correlation between genotype and phenotype, that's it. It's almost diagnostic. It's like if I handed you a plant and I said it's got a C, did I just hand you teosinte or did I hand you maize? Maize. Because every single maize plant has that C. And no teosinte plant has it. So a single base pair mutation, change from a C to a G, or a G to a C rather, depending on which direction you're looking at this, is actually responsible for this change in phenotype. So let's see what that actually does. Time to engage your brain a little bit more. So there's that mutation. At position 18, nucleotide 18, which means where's this mutation happening? It's in the plus numbered nucleotides. It's not in the minuses. So where's this mutation located? What are the negatively numbered nucleotides in a gene? Non-coding, promoter, non-coding, upstream of the start codon, the start site rather of transcription. So what's a plus nucleotide? It's either going to be, what is it? It's probably going to be in an exon. It could be in an intron, but it's going to be somewhere in the transcript, plus nucleotide. So at plus 18 here, there's a G to C change. And that causes what's in teosinte is a lysine, amino acid K, to N, asparagine. So there's a single amino acid change. G to C. So you find the codon. Teosinte is a lysine that has a G. Where in the codon table do you find a lysine that's got a G in it? Where's lysine in the first place? So here's lysine. We've got two codons that can encode lysine. What happened? Where's the mutation? Right, so it's a G to C, so teosinte has a G, and that encodes lysine. So where's the C that encodes asparagine? Right there. So a single mutation at the third position in the codon, from a G to a C, caused one amino acid change in teosinte, and that led to this transition from the closed fruit cups to open. Single mutation. That's all that was needed to start the process that allowed humans to domesticate corn. One nucleotide change makes a pretty big difference in phenotype, the shape of the fruit. Now, if I went into the laboratory and did this, made a single mutation, single base pair change, would that count as genetic modification? If I did this in the laboratory? If I took teosinte from the wild and I just made this one point mutation, I stuck it back out in nature, would you accuse me of doing something unethical or immoral? What do you think? Gray area? because scientists do this. But tomatoes. Tomato fruit size is a complex trait. It means that multiple genes are involved in deciding whether or not a fruit is going to be really big or really small. So in this case, I showed you earlier this picture of the tomatoes on the left. Big tomato, small tomato, beefsteak, and a cherry tomato, plum tomato, something like that. The difference between the sizes of those two fruits, they differ by basically one allele or one gene. And the effect of that gene is the change in size you see from minus to plus. So you add this mutation to tomatoes, you get a detectable but very, very, very tiny reduction in fruit size. So to explain a difference like you see here on the left, how many different genes do you think have to be involved to make that sort of a difference in fruit size? A lot. That's why this is, as we've talked about earlier in the term, a quantitative trait. It's not that you have either a big fruit or a small fruit, and there's one gene that causes that difference in size. There are many, 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 many mutations altogether, or genetic differences, that change fruit size in subtle ways. So is it possible to genetically engineer a tiny cherry tomato? I mean, how much work would it take to do this in the laboratory? If it takes, say, 8 genes. This is just a guess, by the way. This is not fact. What if it took 8 genes to make a difference, that difference in size? Is it worth genetic engineering? Is it worth going to the laboratory and trying to engineer this? What do you think? Is it worth our time? Let's skip the salmon for now. But genetically engineered to express its own gene. Let's not skip it. Does this count as genetically modified? It has no genes from other species. Scientists have just turned up the expression of its own growth hormone. So it produces more of its own growth hormone. Reaches maturity faster. Farm to table faster. Good for humanity. Feed more people. What do you think? Is that genetically engineered? Somebody have an opinion. Is it genetically engineered?
I think it is, but it's not like the ones where people are like, oh, people who don't like GMOs, I don't think they don't like GMOs for these kind of things.
Okay.
I think they don't like it because like.
So I've chosen a bad example. That's
Yeah,
fine.
like when they put like pesticides in the food, I think that's what they're protesting against, or at least for me.
Okay.
I think that's good. Having more fish, that sounds like a good idea.
Okay. Okay. Anybody opposed to that? What's one of the big concerns that people talk about with genetically modified organisms?
Transgenic escape.
So escape. So what happens if this fish goes and mates with a wild salmon? And then you get super salmon, right? These huge behemoths roaming the oceans, and they try to swim upstream to spawn, but they block the whole waterway because it's but that is absolutely a real concern. So what if you put a transgenic organism in nature and then it spreads whatever gene you put into it to natural species or natural populations? Would that be bad? We can't recover necessarily from a situation where we've put a transgenic organism into nature and it overcomes all of its wild type siblings or ancestors.
On the putting the pesticides in the fruit, one of the problems with that is when the bugs, it's supposed to ward off bugs, but then you have the problem of getting super bugs, where the bugs actually use the pesticides. You have to use even more pesticides, so that's another.
So then we get sort of an arms race scenario.
Yeah.
So you put pesticides, it's the same as Roundup and weeds. Weeds evolve resistance to herbicides, so we need new herbicides. We put more herbicides on the plants, and they evolve resistance to the herbicides. Absolutely.
And then the people who are working on
Pardon?
the people who are faking.
And then we have to worry about the people that are working with the plants. So if you make a transgenic plant that has pesticides built in, is that dangerous to other species? So there could be a huge amount of regulatory involvement that needs to go on and a lot of testing before any of this actually gets put into the field or to market, certainly. Arctic apples, another great example. No genes from other species. These scientists from Washington State just turned down the expression of polyphenol oxidase, which is the enzyme that causes browning when you cut an apple. So McDonald's loves this, presumably. Not picking on McDonald's, but you go to you might not know. It's been a while probably last time you had a Happy Meal. But when I go on a road trip and my kids want to eat McDonald's and we get the Happy Meal and they get the little box and the toy and the little tiny hamburger that used to seem like it was really big, right? They get a bag of apple slices. And McDonald's doesn't want to have to throw out the apple slices every day because they turn brown as soon as you cut them. So you buy an Arctic apple, where all that the scientists have done is engineered these apples to produce less of an enzyme they already produce, they just make less of it so that they don't brown as quickly. It's transgenic. It's genetically modified. Somebody did this in the laboratory. But does this really count as genetically modified? Could we, theoretically, just thought experiment, could this have been done by selective breeding? Could this same result have been achieved through traditional methods? Possibly, probably, maybe. What would have to happen to grow an apple that has reduced polyphenol oxidase expression? It would take a lot of time to do what? You're on the right track. So it would take, right, so how long does it take to breed apples? It takes a long time. But what are we waiting for?
Mutation.
Say it louder.
Mutation.
Right, we're just waiting for the right mutation to occur that happens to, by chance, turn down polyphenol oxidase expression. Right, a mutation in a enhancer, for example. Right, one base pair change. Now a regulatory factor doesn't bind there anymore. And so there's less polyphenol oxidase expression. Sure. It's totally reasonable that this might have, probably did already happen in nature in fruit varieties. Nobody noticed because there are so many apples and nobody was looking. Does that matter in our assessment of whether or not something is genetically modified? I'm sure apples in nature right now exist that have lower amounts of polyphenol oxidase expression than others, naturally, just through random mutations like has occurred since the dawn of time. So if scientists do this in the laboratory, instead of waiting for random mutations to occur, is that better or worse? What do you think? Does it matter if a result is achievable through natural means, we just accelerate it by doing it in the laboratory? Does that impact how you feel about genetically modified organisms? Perfect. So, before you go, I'm going to start another quiz. It's going to ask you exactly the same question. I just want to see if anybody's mind has been changed or not, that's all. And of course, this is worth some participation points for the day as well. So go ahead with this. And the next time we get together on Wednesday, we'll continue our conversation. And I won't be able to do office hours today, so if you do have a question or wanted to stop by, I'll be there on Wednesday, or email me, we can set up another time to talk.